Hello, everyone. Thank you for joining the webinar of today titled Safety and Compliance in Herbal Products, Megatrends and Overview, presented by Neotron. I'm Luca Vogesi, Head of Marketing and Business Development in Neotron, and I'm your host for today's session. So if you have any questions in the middle of our presentation, feel free to use our Q&A section or chat box. We will address those questions towards the end of our program. Before we dive into the main agenda of today, let's introduce our speakers for today's agenda. Okay. So here you can see in the screen the agenda of today. So this means introduction of the market, analysis, compliance, and safety analysis packages, herbal dietary trends and supplements, approach the method selection for herbal supplements, and a few case study presents by our colleagues. Then obviously there will be also the question and answer session. So as I was saying, let's focus now on the speakers. First of all, we have Massimo Veronesi. Massimo graduated in pharmaceutical chemistry and technology. He has been working in Neotron since 2012. After a period in R&D department, he became the department head of nutritional. So he deals every day with analysis for vitamins, for amino acids, fatty acids, polyphenol, and many analyses also for the herbal supplements and raw materials. Nowadays is Massimo also the product specialist for the dietary segment of Neotron. And the next up, we have Eva, top two. Eva is graduated in pharmaceutical chemistry and technology. She has worked at Neotron since 2016. And also after a period in R&D, she moved in the GC/LC nutritional departments in 2018. Now is the deputy leader of HPLC SA since 2021. So, just to give you a little brief introduction about Cotecna. Cotecna is a leading provider on testing, inspection, and certification services. We offer a solution to make supply chain safer and more efficient for our clients. We are a Swiss-based company headquartered in Geneva with offices and labs across the globe. Today, now we have over than 7,300 employees with 140 offices and labs. Let me say that our network of trusted professional and certified labs provides expertise across the 5 sectors, namely agriculture, food, safety, metals and minerals, consumer products, system certification, and SGS solution. I'm also proud to communicate to you that Cotecna is celebrating the 50-year anniversary. So please follow us in our social media channels for more information and also on the milestone for the past 50 years in the testing, inspection, and certification services. Let's now go on. So about Neotron. Neotron is a leading providing testing company based in Italy. We are in Modena. And as you can see here, is a lab that has more than 500 employees, technicians that work in lab, technicians that can support you. With 2 different building sites, we receive more than 1,000 samples per day, having more than 800 meters with plus 3,500 accredited tests. So we could be the right partner for your needs. So going on, we can now go directly for the first topic. So for the first one, I leave now the words to Massimo and to his contribution on the webinar of today. Please, Massimo.
Thank you, Luca. Hello, everyone, and thank you for joining us in this webinar focused on compliance and safety in herbal products. I will begin with a general introduction on herbal supplements, followed by market analysis. Afterwards, I will discuss our analytical packages regarding compliance and safety, and finally, highlight some of main herbal trends. A herbal product, also known as a botanical supplement, is a dietary supplement comprised of one or more herbs that provide the defining benefits and claims of the product. For centuries, a multitude of plants and herbs have been used for their therapeutical qualities. The incorporation of these botanicals into dietary supplement is a prevalent practice within the industry. A rising trend towards holistic wellness is evident, marked by a rebirth of natural remedies. People worldwide are more and more turning to plant-based alternatives to address diverse health concerns and promote overall well-being. As consumers' interest in wellness grows, brands are continually innovating with new products and new formulations to adapt to the evolving needs and preferences of the population. For this reason, botanical ingredients are among the fastest growing segments of the dietary supplement industry. Next, please. As a significant portion of consumers incorporates one or more dietary supplement into their diet, herbal products play a crucial role in the global market. The herbal supplement market size was valued at $11 billion in 2022 and is projected to reach $21 billion by 2032, growing at a CAGR of 7.1%. As we can observe from this graph, Europe is predicted to be the fastest growing region. The level of innovation within the supplement market is notably high, as numerous companies are continuously creating new products and new formulations, each featuring its own distinctive blend of botanicals, vitamin, amino acid, and so on. The rapid growth of this sector requires all testing laboratories to develop and optimize new methods suitable for analyzing these products. Regarding the herbal topic, Neotron offers comprehensive compliance and safety analysis for raw materials, intermediate or finished product throughout the supply chain. We can offer various solutions to ensure the compliance with raw material and finished products, as well as prioritize safety, which is paramount for guaranteeing raw materials and ensuring consumer safety when purchasing the final product. Depending on specific customer needs, the Neotron team is able to offer a guide in choosing specific assays relating the active to be identified and quantified. For compliance testing, we can offer a specific or specific method. Here, there are indicated some of the aspecific methods. We are able to measure the concentration of an entire class of compound. This technique provides an accurate, easy, and quick result. They are especially useful to determine the sum of an entire family of analytes that form an ingredient active part. At contrary, if the target is the quantification of the single analytes, we have at our disposal a whole range of methods for the determination of the active compound. This is a quick overview of the analysis we can perform in our laboratories. There are all in-house methods developed and validated for specific products and then extending to others. Many of these are also accredited. For example, the glyceric acid in licorice is an accredited method for these specific botanicals. Focusing on some target, for example, catechins, anthocyanins, and procyanidins, we can perform the HPLC UV method for high content products where we can determine all these groups of compounds singularly. The same approach is adapted also to other analytes like caffeine. In product where it is present as a natural source or is added as an ingredient, we can perform the analysis by HPLC UV. Instead, for traces, or in case we have to prove the absence of this compound, we can determine caffeine in LC-MS. Many of these compounds are inherent to the phytochemical profile of herbs and serve as a key element for the authenticating and controlling the quality of botanicals. These chemical markers represent a biologically active component of the plant, often unique to specific botanical types and present at specific concentration. Assessing these chemical markers can serve as a valuable method for verifying the quality, efficacy, and safety of botanical ingredients. To mention one, berberine. Berberine is the primary alkaloid of Berberis vulgaris, has demonstrated a range of health benefits such as inflammation reduction, enhanced blood sugar regulation, and antibacterial properties. This herb is used as an active ingredient in the manufacturing of dietary supplements. The identification and quantification of berberine in both raw materials and finished product serve as a crucial quality control measures for producers in this industry. In Neotron, we can perform the analysis by HPLC, either in raw material and a big variety of finished products. Moving on to another important aspect of quality control, which is safety. Plant-based supplement safety is the result of an integrated set of factors, such as suitable production structures, the adequacy of the system, and effective control of the process. The purpose is to identify possible risk factors and intervene promptly to restore normal condition. The producer will want to assure that their product is not contaminated by micropathogenic organisms, by chemicals or physical contaminants, because this guarantees the quality of their supplement. In this slide, we report the main test performed by Neotron as part of the safety evaluation. For example, Neotron Microbiological Laboratory can support its customer with specific analytical packages relating to microbiological controls. Instead of focusing on the chemical controls of residues and contaminants, there are some of the Neotron method we can conduce in a wide range of products. For example, for pesticide. For pesticides, we can propose different analytical packages from simplified to more extensive packages which are continuously updated with new regulation alerts. But we have also a list of pesticides that follow pharmacopoeia standard and specification. Now, let's shift our focus to the key market trends we have identified, particularly emphasizing the most popular and in-demand dietary supplement, and delve into how our quality control measures align with these trends. In the past years, there has been an increasing interest in rosemary extract, as you can see from the slide. Rosemary is known for a multitude of benefits and therapeutic effects, such as antioxidant and anti-inflammatory effect, improves memory, and helps boost immunity system. It also stimulates hair growth, and recently it used as a raw material in cosmetic industry. Rosemary properties are related to its main constituents, which are rosmarinic acid, carnosic acid, and carnosol. We can perform the assay of these compounds in HPLC UV, in raw material and finished product. In case of traces, we have also a method in LC-MS. Another big trend is ashwagandha, with its active compound vithanolides. Ashwagandha is a small evergreen shrub that grows in India, Middle East, and some part of Africa. Its botanical name is Withania somnifera, and also known as Indian ginseng, which is widely incorporated into supplement due to its myriad benefits, aiding in stress reduction, insomnia management, and enhancing brain function and memory. Here, are some data market regarding ashwagandha. And with Neotron, we can test the compliance in raw material ashwagandha root by determining the vitanolide A in HPLC UV. Vitanolide A is the primary component among the vitanolides. Another herbal trend that has been used for centuries in traditional medicine for treating several health conditions is Sanjonwort, or Hypericum perforatum. It is a plant belonging to the Gutiferae family, quite widespread in Europe. Its flowering tops are rich in bioactive compounds, such as hypericin and hyperforin. It is used in various industries, such as pharmaceuticals, cosmetics, and dietary supplement for different purposes. To mention some, such as helping with mood disorders and overall mental well-being. The demand of Sanjonwort is expected to experience substantial growth in the coming years. In Neotron, we can perform the identification and quantification of hypericin either on the raw material and in finished product by HPLC UV. Lastly, it's important to highlight the growing trend in incorporating turmeric into a whole array of dietary supplements. Curcuma longa, commonly known as turmeric, is widely utilized for its antioxidant and anti-inflammatory properties. It's valued for supporting joint health, digestion, and boosting immunity system. The major bioactives of turmeric are curcuminoids that include curcumin, dimethylcurcumin, and bis-dimethylcurcumin. In Neotron, we can perform the determination of total curcuminoids expressed as curcumin in HPLC UV. With this method, we can identify the composition of each curcuminoid. But curcumin is also used for food coloring. For these cases, we can perform the analysis in HPLC UV or in LC-MS, depending on the concentration. This was a comprehensive overview of our offer in the matter of compliance and safety and analytical packages. Now we will further focus on some of these methods through specific cases regarding our analytical approach, which will be discussed by my colleague Eva. Thank you.
Thank you, Massimo.
So
Hello, everyone.
thank you, Massimo. Thank you. As you have already seen, we have a lot of different analytical proposal in Neutron, always align to the last updates and trends of the market. As Massimo said, now we leave the words to a more concrete and real case study, let me say, overview made by Eva. So please, Eva, it's now your turn.
Hello, everyone. My name is Eva, and I will discuss our analytical approach to selecting the most suitable methods for quality control tests, following with an in-depth look of two case studies. A pivotal aspect of ensuring quality control is selecting the appropriate analytical method. The choice depends on different factors, including the chemical properties of the target compounds, their concentration in the sample, and the technology utilized in the product. An analytical method used for raw material may not always be appropriate for determining the compound of interest in the finished product, which can exist in various forms like tablets, capsules, soft gels, powders, and each product has a unique composition of active and excipients that can impact the extraction and the detection of the analyte we aim to quantify. Frequently, we encounter ourselves in scenarios where adjustments to the extraction or detection method are necessary to accommodate to the unique characteristics of the product. To ensure the quality control of both raw materials and finished products, there are some approaches that we can employ. For instance, If a product adheres to pharmacopoeial monograph standards, also known as simply monographs, we can conduct the necessary tests outlined in the pharmacopoeia. Pharmacopoeias are elaborated and revised by groups of experts and working parties and provide tests, methods, acceptance criteria to define the identity, the composition, and the limits of contaminants of a specific product. They serve as a transparent and publicly accessible standard methods and provide the essential specifications for dietary ingredients and finished dietary supplements. They offer a shared understanding across the supply chain regarding the necessary quality attributes for products and their ingredients. However, they come with various specifications to comply when performing official tests. For instance, if we aim to test, let's say, a 3% dry extract by using an official method with an acceptance criterion of not less than 5%, we cannot apply the assay due to the failure to meet the specifications. Another aspect to consider is that the official texts primarily offer assays for raw materials, with less information available for finished products. Nevertheless, monographs are continually expanded and revised with new tests and analytical procedures, so it's advisable to refer to the official pharmacopoeias whenever possible. We can resort to the USP that provides more than 300 monographs for botanical dietary ingredients and dietary supplements. Or we can consult the European Pharmacopoeia, where are listed more than 2,000 monographs for herbal drugs. Here are some examples of the monographs that we perform in Neotron. For example, for soya lecithin, we can utilize the USP monograph, which assesses the phospholipid content serving as the primary biomarkers for this particular material. For dry extracts of ginkgo biloba, we ascertain the presence of active compounds such as ginkgo flavonoids, ginkgo terpene lactones, and perform additional tests by referring to the European Pharmacopoeia. Another monograph worth to mention is the powdered bacopa. According to USP on the raw material, the compliance is established by determining the content of the bacoposides. An alternative way for compliance testing of an ingredient and finished product is the feasibility and the development of a method for that specific product. In our R&D department, multiple tests are conducted to define a suitable extraction and detection method. When selecting the extraction method, various steps are involved in defining the most suitable one. We must consider factors like chemical and physical properties of the sample and the complexity of the product. The sample could be simply dissolved in a solvent, or a more complex extraction is needed involving boiling, distillation, SPF purification in order to separate and isolate the target from other components and obtain a clear extract. Similarly, the selection of the separation technique depends on the chemical properties of the analytes. We can either opt for liquid chromatography or gas chromatography based on the characteristics of the compounds. Lastly, the selection of the detection method relies on one or more properties of the compounds, such as fluorescence, ultraviolet absorption, volatility. Another crucial aspect to consider is the concentration of the target in the sample. If the target is present in high levels, we can utilize, for example, the UV technique. However, for low concentrations or trace amounts, we must employ the LCMS method, which is a highly sensitive detection technique. Furthermore, the composition of the product also affects the selection of the detection method. Despite UV offering a good resolution, if there are interferences in the sample or insufficiently separated multiple compounds, UV detection may not be suitable for identifying our target. In such cases, resorting to LCMS detection is necessary. A significant amount of bibliographic research and testing is conducted to establish the suitability of a method for a dietary supplement. Once the method is developed and ready to use, we can proceed with a method validation for this specific product. In this step, several parameters are evaluated, such as specificity, reputability, accuracy, intermediate precision. All these parameters contribute to the method's robustness. After developing and validating the method, we can test its feasibility in other products containing our compound of interest by conducting feasibility tests. An interesting example of method feasibility is ergosterol. Ergosterol is a precursor of vitamin D2 and is present in different species of yeast, mushrooms such as shiitake mushrooms, lion's mane, and lately is a popular topic in dietary supplement sector because of its potential health benefits. In Neotron, we have an in-house method for years now to determine ergosterol in products other than supplements. And recently we received a request from a customer for testing ergosterol in mushrooms used as a raw material. So what was our approach? Firstly, we wanted to test the feasibility of the actual ergosterol method in this new product. And the method turned out to be suitable without requiring substantial changes. The samples were tested in duplicate to ensure the data precision, and we obtained a good reputability. Also, recovery tests were conducted in order to evaluate the performance of our method, and we achieved positive outcomes. Following the feasibility in the raw material, we determined ergosterol in the finished product. The finished product consisted in tablets containing the mushrooms previously tested as the active ingredient. The method proved to be suitable also for the finished product. The last step was the method validation for the raw material and the tablets by following a specific protocol established by our quality department and approved by the customer. A different scenario is the arbutin case, where changes of the actual method were needed in order to test a new product. Arbutin is a hydroquinone present in a variety of botanicals, including beerberries, cranberries, sweet peas, and pears. However, another application of arbutin as a biomarker is in the detection of juice-to-juice adulteration. A common practice involves using pear juice to adulterate apple juice. Given that pear juice is sweeter and less expensive, it has a low commercial value. Studies indicate that pear juice contains arbutin in specific concentrations. Meanwhile, its presence was verified in trace amounts in apple juice. In Neutron, we have a method for testing arbutin in supplements and cosmetics by HPLC UV. There was a request to determine the arbutin content in apple juice, a product which was never analyzed before. We tested the feasibility of our method developed for supplements in the apple juice. However, we obtained interfered chromatograms, making it impossible to identify and quantify our target. This was either due to the composition of the product and the levels of concentration of arbutin, which were quite low. We couldn't reach that sensitivity with a UV detector. So how did we overcome this issue? By developing a method in LCMS. Arbutin's transitions were identified and the chromatographic and extraction method were optimized. And through these efforts, we were able to enhance the method, obtaining a clear chromatogram that facilitating the identification and the accurate quantification of the compound. So having an alternative method in LCMS is beneficial for samples with a significant interferences amount and for confirming the results obtained via HPLC-UV. In conclusion, the main emphasis is on three crucial points. Firstly, our commitments to providing comprehensive analytical packages ensures that the herbal finished dietary supplements and ingredients meet the highest standards of quality and safety. A range of offerings can be tailored to specific needs and assure that every aspect of the product is thoroughly evaluated and controlled. Secondly, the choice of analytical method is paramount in ensuring effective quality control. Our expertise and guidance in selecting the most suitable methods for dietary supplements guarantee accurate and reliable results. Neotron offers support in every step of the way, ensuring that the products meet certain requirements and exceed expectations. Lastly, in a rapidly evolving market, adaptability is key. We are able to offer feasibility studies and develop custom methods for new and specific products according to customers' various needs. Thank you so much for your attention. Hope you enjoyed the presentation, and now I leave the stage to Luca.
Thank you, Eva. Thank you very much. And now we are going to move, let me say, in the question and answer session. So I think that we received some questions or by chat, but also by the Q&A session dedicated field. So we are going, I think, in the first question received. So the first one is, in both presentation, you emphasize the importance of food safety in terms of food fraud. What can Neutron propose to achieve the increasing demand of food monitoring and authenticity check? So I think that this could be a question for you, Massimo, probably that you can reply, correct?
Yeah, okay.
Perfect.
Food safety is a matter of global importance. As Neutron, we are actively working together with our customer to identify and mitigate the risk of food fraud. We can propose different analytical methods meeting the European Commission report and the Codex Alimentarius requirements. Chromatographic techniques enable the detection and identification of chemical markers for food authentication. However, qualitative DNA-based methods are required. The use of next-generation sequencing, or NGS, is an emerging approach that allows the detection of multiple species in complex and processed foods. For example, the analysis of cinnamon adulteration could be a good example of how these two techniques can merge. Due to its high cost through cinnamon, that it is the cinnamon verum, which is the only produced in Sri Lanka, it is adulterated with cinnamon cassia due to its low commercial value. On the other hand, cinnamon cassia contain a high content of coumarin, more or less 1%, while in cinnamon verum is present only in minimal amount. We are around 0.04%. This value can be calculated using HPLC-UV. But to confirm this data, the NGS technique, through its screening evaluation, gives an identification of the botanical species present in the sample.
Thank you, Massimo. Thank you. So we can go through the second question that we have received. So Regarding compliance, the stability of the analyte in product play a crucial role. What's Neotron approach on this issue? I think that could be managed also this one by you, Massimo.
Yeah. Yeah, a stability study is performed to ensure that the quality and safety of supplement are guaranteed until the expiry date. At first, we make the feasibility assessment with the evaluation of the overall feasibility of the requested study, and then we develop the analytical offer, including testing, timing, and storage. In a second time, we can store the samples under the provided condition for the entire duration of the study. We perform the analysis according to the method required at each time planned. During the study, we make the verification of data consistency based on acceptance range declared. On time of feasibility assessment, the required timing, testing, and climatic conditions are evaluated from us. When these information are not reported on a stability protocol or generally are not defined by the customer, we refer to the guideline recommendation. We can perform both long-term and accelerated study in terms of testing, frequency, and storage. We have to say that our equipment can cover all the storage conditions defined in ICH guidelines.
So thank you, Massimo, for the answer to this second question. Let's proceed with the other two questions that we have received. So I think that before we go with number 3, it's rolling. They arrives. So we put in the slide as soon as they arrive. So the 3, what are the advantages of having a validated method? I think that this one could be taken by you, Eva. What do you think?
Sure, yes. Having a validated method, for instance, ensures that the results obtained are robust, accurate, specific to the target, with data precision within the acceptable limits. The range of acceptability of these parameters are established during validation, and if there are fluctuations in these parameters during routine analysis, let's say, they can be attributed to the sample's characteristics rather than to the method itself. And this is crucial, for example, in stability studies that Massimo mentioned before, where variable results may occur. A validated method confirms that these discrepancies are not due to the method, but are linked to the product.
Thank you, Eva. Thank you for your answer. So we can go through number 4 in the slides. So can you conduct an accreditation for herbal dietary supplement?
So the accreditation of herbal supplements as a generic category is complicated. As mentioned in the presentation, they come in different forms, tablets, capsules, gummies, liquids, powders, each with different characteristics and a different composition of actives and excipients. For this reason, it's not always possible to apply the same analytical method to all types of formulation, even if the target compound is the same. So, what can we do instead is a product-specific validation, also called a method verification, by following a protocol established by our quality department where different parameters are defined and monitored. For example, we can define the reputability by performing the analysis, let's say, 6 times, or the specificity where we establish if our method is suitable for detecting our target. These and other parameters define the robustness of the method for that specific product.
Thank you, Eva. Thank you. So we have some other more questions that I received. So the first one, I think that I can take it. because it's something that is more related to the work that we are currently doing with also our colleagues of Cotecna is in this historical moment, this comes from the chat and the Q&A session bar. In this historical moment, is the search of new supplier of raw material is a key factor for industries in this sector. How Neotron can help company with this topic? So I'm taking this mind because it's, as I said, strictly related with the services that we can also perform with our colleagues of Cotecna. So Neotron at the end remains an analytical lab, so we perform analysis. But I can say to you that in this moment, many companies try to reach new producer of raw material because they are very new in the market, this raw material. But also because there is a lack sometimes of this raw material. So the search of new supplier is always a very key point and important point for many companies. So in this sense, I can say to you that if you are searching for new companies of raw materials and then you are interested in qualify it and see if you have a good quality of the raw material from these suppliers, we have the possibility through our colleagues of Cotecna that are present all around the world, for example, to perform for you audits and pre-shipment services, what we call end-to-end services. So this means set up audit checklist in ISO or in GMP related to the site that you want to inspect of your suppliers, but also related to the raw material that you are going to, so the herbals that you are going to buy from them. And then the second step will be the pre-shipment. What we call pre-shipment is a sort of sampling that our colleagues can do before you buy the raw material from the suppliers in India, in China, or wherever is located your suppliers, and send it to us or to you to perform an analytical tests before you buy the raw material. This is something that at the end is very requested from our customers that wants to deal with new raw materials all around the world. So we can, at the end, summarizing my answer, is we can support you 360 degrees from the auditing of new supplier sites and also related to raw material that you want to buy from them, by sampling, so have an evidence before buying the raw material if the COA, the analysis present in COA are compliant or not, and then obviously perform testings also after the pre-shipment because we are analytical lab, so we are skilled on that. So it has arrived another question that I think that I'm going to take. You have talked about compliance in the presentation, so means assays, but in terms of safety, what's Neutron's position about contaminants and in particularly about pesticide analysis? I can take also this mind just to explain a little bit the way of working of Neutron. Obviously, the aim of today's meeting, it was to have an overview on herbals, the trends, what we can do also for compliance in terms of assay, identification, and then obviously the safety is very important also for us. Neutron is a leader testing in contaminants, start about 40 years ago in contaminants and pesticide in food, then baby food. So we have a long expertise also in these complex matrices. And we offer specific packages that could be in compliance with pharmacopoeia, but we have also other specific packages for herbals that are in compliance with regulation, but also renewed continuously. It's a rolling process for us. We have a regulatory office that take care of the updates from the authorities in terms of matrices and limits to be reached, but also an internal group that takes care of the statistics of pesticide and their positivity to continuously update our analytical packages for pesticide basing on our experience. So I can say to you that some of our packages can count more than hundreds of different pesticides that we can propose to you, or eventually reach your questions if you have just done a risk assessment based on your products and you are focused on some pesticide. We can also work on that. So just to conclude, let me say that in terms of pesticide, Neotron is one of the leaders in terms of testing in the market, in the Italian market and in European market. So we can propose to you our solution, or eventually we can receive your list, your risk assessment, and then apply it in our labs. It's 40 years of experience, so it's one of our major, let me say, business. So we are here for supporting you also on that, absolutely. So I don't yet receive any other question right now. So if there are no more questions, let me say you can eventually also continue to make these questions in this chat, or eventually in the specific section related to Q&A. And obviously, we will have the possibility to reply you also in the next days. But let me say that right now we are at the end, at the conclusion of our program of today. In any case, we will send you an email in the coming days containing a survey form and obviously a recording of the webinar. And we will be able also to reply to additional questions. If you are more interested also in Cotecna services, we will also providing you a link for our newsletter subscription. Okay. We hope that today webinar was beneficial for everyone in terms of scientific divulgation, but also for the business of you and your company. So at this point, I would like to have a big thank you to our speakers, Massimo and Hiva, for sharing their expertise today, to all of us. And have a great day, everyone.
Thank you.
And see in our next webinar. Thank you.
Thank you. Have a nice day.
Thank you. Bye.
Bye.